from Haifa University.
So hi, my name is Ashwani Choudhary and I am a PhD student in Dr. Sunny Stern's lab in University of Haifa. And I'm going to talk about the early hyperexcitability phenotype which we have observed in the iPSC-derived cortical neurons from the patients which had certain ASD-related mutations. So before I start the talk, I would like to give an overview. I will speak about a bit of ASD, although the audience is aware about the ASD. And I will also speak about the early hyperexcitability phenotype, which we observed in two kind of mutations, ASD-related mutations, which is green 2B and SHANK3 mutations. And lastly, I will also talk a little about some of the exciting data we have about the mechanistic insight about how this hyperexcitability is transferred among the cells. So autism spectrum disease is one of the complex disease, and it is a polygenic disease with high heritability estimates. And according to the DSM-5 characterization, we know that it is because of the abnormal, the patients are diagnosed with abnormal social interaction, repetitive behavior, and also delayed language skills. So as compared to other neuropsychiatric disorders like schizophrenia or bipolar disorder, autism spectrum disorder manifests early in childhood. And if you look at the prevalence level, according to the WHO estimates, the prevalence is around 1% worldwide. And again, we have already heard it in the sessions that the boys are more affected than girls. And some of the countries have high rates, like USA has 1 in 44, Israel in 1 in 78, and India and China, which was updated, which has old data, says that it's 1 in 100. So if we look at the genes associated with ASD, in the morning we had a talk about the genetics of ASD, and we came to know a lot about the genetics of ASD. And what the Simons Foundation has initiated two kinds of research programs like SFARI and the SPARK, and they have contributed to our knowledge of the genes affected in ASD. And there are two genes which are important and which I'm going to talk about today is SHANK3 and green 2B. Now green 2B is among the high confidence genes and high confidence risk genes of ASD, and also SHANK3, but they are not specific to autism. They are also involved in other kind of disorders like developmental delay, intellectual disability, and also SHANK3 is also known to be associated with Phelan-McDermid syndrome. So a little bit about the functions of SHANK3. If you look at the SHANK genes, these are important genes which are which have a synaptic functions. They act as a scaffolding protein, and they actually helps in the scaffolding of the glutamate receptors onto the with the cytoskeletal elements. And it was first implicated in neurodevelopmental disorder, as I said, in Phelan-McDermid syndrome. So this makes it an important protein for neurotransmission, especially in the excitatory neurotransmission. And similarly, green 2B is another protein which is part of the NMDA receptor. And if you look at the crystal structure of NMDA, it has two green 1 and two green 2 subunits. And if you look at the simplified diagrams, you can see that there is a amino terminal domain, the agonist binding domain, the transmembrane domain, and C-terminal domain. I'm explaining this because we have used some of the mutations in the agonist binding domain of one of the patients which we have derived from. So the iPSC lines, so we know that there are models of various models for ASD, but iPSCs has a bit edge over them because we are having the same genetics as the patient, and that way we can understand the cellular and the molecular pathways much better than other model systems. And so if you look at the green 2B, the green 2B gene, the mutation was found, it was a female child which had a mutation, missense mutation, which led to the point mutation of G to T at a particular position of exon 11, and which led to the mutation at agonist binding domain. At the same way, if you look at the SHANK3, it was a frame shift mutation. Both are loss of function mutation, But in the SHANK3 mutation, the mutation was at the exon 21, which is at the proline-rich region, which helps in the binding of Homer to the protein. So we thought of doing the cortical neuronal differentiation. Again, as we heard in the morning, like cortex is the major area which is affected in ASD, and if you look at the expression of green 2B and SHANK3 in human protein atlas, and if you try to, you will see that the cortex is the region where it is highly expressed, both of the genes. So we did a cortical differentiation, where we followed a particular protocol where we do we make EBs from iPSCs, and then we make rosettes, then we make NPCs, and then we do a differentiation for around 4 to 5 weeks. So these are some of the representative images of the iPSC lines from which we created 3D cortical EBs. From there we made NPCs, which were stained for the neural progenitor markers like nestin and PAX6, and then we also differentiated them into mature neurons which expressed MAP2 and TBR1. TBR1 is one of the markers for cortical neurons. So we knew that our neurons are cortical. So using this, we differentiated the green 2B neurons into the cortical neurons, and we found that the green 2B neurons, at the 3rd week of the differentiation, they were hyperexcitable. The hyperexcitability, when we say hyperexcitability, what I mean is there was a higher sodium current also, and plus there was higher total evoked potential, which was also not only, this as compared to the controls, they had they were more mature, and they were more sensitive to the current injection. So if you we also looked at the EPSC rate of these cortical neurons of the green 2B lines, and we found that the EPSC rate was also high in the patient cortical neurons as compared to the controls. So similarly, in the SHANK3 mutation also we found higher sodium current and total evoked potential, and also higher EPSC rate. So this pointed that these mutations, although they had a different genotypes and derived from a different patient, from female patient, they exhibited similar phenotype of hyperexcitability. So thinking about the mechanisms, we stumbled upon exosomes. Exosomes, which are known to be are mediators of cellular communication. They are membrane-bound lipid membrane-bound extracellular vesicles, and they can be produced from any eukaryotic cells, and they are known to have both positive functions like neuroprotective effects or the transmission of protective factors between the cells, or they have some negative functions like they can also do the immune infectious, they can spread the infections, and they can also lead to the waste removal from one cell to another. So these exosomes are basically produced inside of our eukaryotic cells, and the contents of the exosomes are quite similar to the source cells they are produced from. So initially the early endosome particles, they fuse to become multivesicular bodies, and from the multivesicular bodies, they again they release out from the cells to form extra exosomes. So exosomes have been initially shown to have a therapeutic potential in ASD mice models, also from a Tel Aviv University group from Daniel Offen, and also from some of the groups from China. So we thought that exosomes could have some effect in ASD-based models, our models, and we used our SHANK3 models for this particular experiments, and we isolated exosomes. So exosomes, for isolating the exosomes, we needed to, because exosomes are of particular size, like 30 to 200 nanomolar size, they are, so we need to clear of larger particles, extracellular vesicles, like the dead bodies and the apoptotic bodies. So we did a centrifugation at different speeds, and then we isolated the condition, and then we cleared the conditioned media, and we did the ultracentrifugation to get the exosomes, and we treated those exosomes on the SHANK3 cortical neurons. But what we wanted to look at was the effect of exosomes on the control and the patient line, but we switched the exosomes from control to the patient and patient to the control to see what are the effects and what kind of molecules it could produce, one cell could produce to and affect the other cells. So when we transferred the, when we did the exosome switching experiment and we did the electrophysiology, so we found that the sodium and the potassium currents were higher in, sodium and potassium currents were not much changed in, although control treated or patient treated with the control exosomes or control treated with the patient exosomes neurons, but the control and SHANK3 was, as observed earlier, it was this, they had some difference in the sodium currents. But when we look at the hyperexcitability, we found that the control treated exosomes started to behave like the patient cortical neurons when they were treated with patient neurons, but the patient neurons were not much changed. So this was also when we looked at, we did a specific analysis of the spike widths and the spike threshold. We found that there was a change in the, even in the spike width of the total evoked potentials, and also, but there was no change in the spike threshold. So we also measured the EPSC rate and of these neurons, but the EPSC rate, we didn't find any change in the control treated neurons with the patient exosomes. But again, the patient neurons with the exosomes remained the same. So we also looked at the spontaneous firing rate of the cortical neurons in these exosome treated cortical neurons, and we found again that the control treated neurons were behaving like the patient neurons, and but the SHANK3 treated mutated neurons did not change, alter significantly. So this showed that there is a transfer of some kind of molecules which are causing hyperexcitability, which is responsible for hyperexcitability and which can be transferred via this exosome switching. So we hypothesized that there could be some molecules which are thrown out of the cells which could be causing this hyperexcitability phenotype, and we are still trying to look at those mechanisms. So the conclusions are the cortical neurons from ASD iPSC lines like SHANK3 and green 2B became hyperexcitable at 3rd and 5th week, green 2B at 3rd week and SHANK3 at the 5th week of differentiation, and the exosome switching showed that the transfer could be done from the early hyperexcitability phenotype from one cell to another, which means there are some diffusible molecules which are being transferred by exosomes. And we can also conclude that these ASD-specific mutations actually cause early maturation and hyperexcitability phenotype in these two different kind of mutations which do not have the same genotype but have same converging phenotypes. Thank you. Before I conclude these works, this work was not possible because without my PI, Dr. Sunniston, who has thought about this idea and who has helped me with most of the experiments, and Idan Ross, who did most of the electrophysiology experiments, Yara, who did the electrophysiology analysis, and Utkarsh has also helped in the electrophysiology. And also the lines were provided by Dr. Tali, who is from Amek Medical Center, and also there was we got technical help from Dr. Gadvatin's lab, and the patients, and also from Dr. Ronan Spiesel from Clalit Health Services. So thank you.